Silence. Darkness. And then, a war begins. 250 million, that is the number of competitors entering the most brutal biological race in all of nature, yet only one will survive, and this is not luck, this is not chance, this is molecular combat, and the rules are more complex than anything the human mind has yet fully understood. Inside the human body, invisible to the naked eye, a selection process is underway that took 3 billion years of evolution to perfect, a process so sophisticated, so ruthlessly precise, that modern science is still uncovering its secrets, and what you are about to witness is not a simple journey, it is a gauntlet, a test of strength, chemistry, intelligence, and molecular identity. In the male reproductive system, a remarkable manufacturing process never stops, because every single second 1,000 sperm cells are produced, each one a marvel of biological engineering, a microscopic vessel carrying half the genetic blueprint of a potential human life, armed, programmed, and released in numbers so vast that the scale is almost impossible to comprehend. Their mission is singular: find the egg, a single cell no larger than the period at the end of this sentence, hidden deep inside the female reproductive system, and the distance they must travel relative to their size is the equivalent of a human swimming nearly 30 kilometers upstream through an obstacle course designed to eliminate the weak. The first challenge awaits immediately at the cervix, to the naked eye a narrow passage, but at the molecular level a fortress, a biological wall engineered by millions of years of evolution specifically designed to stop the unworthy, because cervical mucus is not simply a fluid but an intelligent molecular mesh, a three-dimensional biological filter with properties that actively change based on the time of ovulation, and the wrong shape, the wrong movement pattern, the wrong molecular signature on the surface of their heads means the mucus locks them out permanently. Watch closely as sperm with abnormal tails spin in place and die. Sperm with malformed heads are chemically rejected, and sperm that are simply too slow are consumed by the environment before they travel even a millimeter, because in this single passageway alone over 240 million sperm will perish, gone, before the real battle has even started. But how does the cervix know which sperm to let through? The answer lies at the molecular surface of each sperm cell, where embedded in the outer membrane are hundreds of surface proteins, unique molecular identification markers, that the cervical mucus scans like a biological security system, permitting only the healthy, the intact, the correctly formed to pass through. From 250 million, fewer than 2 million remain, and these survivors press deeper into the body, swimming through the warm interior of the uterus, their flagella beating in powerful coordinated waves, being guided by something they cannot yet clearly detect. The uterus opens before them like a vast cathedral of biological architecture, its walls thick, living, and pulsing with rhythmic contractions that are not passive but actively creating currents that push some sperm forward and sweep others helplessly backward, because even here the environment is selecting, even here the weak are being separated from the strong. But the uterus holds a far more dangerous threat. Embedded within its walls and waiting is the full power of the female immune system, macrophages, neutrophils, and natural killer cells, an army of biological warriors whose entire purpose is to identify and destroy foreign invaders, and to the immune system sperm are foreign invaders carrying foreign DNA and foreign proteins, triggering every alarm the immune system knows the moment they enter the uterus. Macrophages engulf sperm whole, neutrophils release toxic chemical clouds that dissolve sperm membranes on contact, and natural killer cells patrol the uterine walls destroying anything that registers as non-self, creating a paradox of biology where the body both needs the sperm and tries to destroy them simultaneously. And yet the species survives, because the egg has a plan. Even before fertilization the female reproductive system releases a carefully calibrated cocktail of molecular signals, chief among them progesterone and specialized immune-modulating proteins, that drift toward the immune cells like whispered commands to stand down. Not completely, but enough to create narrow corridors of relative safety through which only the strongest sperm can pass, a phenomenon scientists call immune tolerance. Where the female body temporarily suppresses its own immune response but only for sperm that carry the correct molecular identity, while continuing to target the abnormal, the damaged, and the genetically compromised. The survivors, now numbering in the tens of thousands, reach a critical junction where the uterus ends in two directions, two fallopian tubes, one leading left and one leading right, with only one containing the egg and the other an empty biological dead end, and the sperm have no map, no compass, no way to know which path leads to their destination, so half will choose wrong and swim into darkness forever. But deep inside the correct fallopian tube, the egg is not waiting passively. It is broadcasting, releasing a continuous stream of chemical signals called chemoattractants that drift outward through the fluid like invisible smoke signals, carrying a message encoded in chemistry. Come, this way, find me. In a discovery that stunned the biological world, researchers found that sperm possess olfactory receptors, the same type of molecular sensors found in the human nose, embedded in the sperm's surface and tail, detecting the egg's chemical signals with extraordinary sensitivity, so that when a chemoattractant molecule binds to a receptor it triggers an electrochemical reaction that physically changes the direction and intensity of the sperm swimming, meaning the sperm can smell the egg, and now it turns toward it with a phenomenon called hyperactivation, its flagella beating faster, its movement more directed and more purposeful than at any point in the journey. But the fallopian tube is not a smooth highway. Its walls are lined with millions of tiny hair-like cilia beating constantly in waves, creating powerful currents that work directly against the sperm, pushing the weak backward and allowing only those generating enough raw biological power to continue forward. And then, after a journey of almost incomprehensible difficulty, they see it, the egg, enormous relative to their tiny forms, glowing with metabolic energy, surrounded by a cloud of protective cells called the corona radiata, and the chemical signal is now overwhelming as fewer than 20 sperm remain from the original 250 million, surrounding the egg and probing its outer layers, carving a path through the corona radiata using enzymes before facing the most formidable structure in human reproduction, the Zona Pellucida, a transparent but incredibly strong glycoprotein shell that is not simply a physical barrier but a molecular recognition system of breathtaking sophistication, containing proteins called ZP3 that act as biological locks which only sperm carrying the precise molecular key can bind to, while all others simply slide off, drift away exhausted, and die. The moment a sperm successfully binds to the Zona pellucida, it triggers the acrosome reaction, one of biology's most dramatic molecular events, where a specialized vesicle sitting like a cap over the sperm's head ruptures and releases powerful digestive enzymes including acrosin, hyaluronidase, and proteases that attack the Zona pellucida's protein matrix, dissolving molecular bonds and creating a microscopic tunnel through which the sperm advances, drilling chemically and relentlessly, molecule by molecule, while multiple sperm drill simultaneously in a race within a race, each depleting its ATP energy reserves at maximum rate, knowing that exhausting those reserves before breakthrough means dying at the wall just inches from victory. And then, in slow motion at the molecular level, one sperm breaks through, its head making direct contact with the egg's plasma membrane, where Zuma-1, a protein found only on sperm, reaches across the nanometer gap and finds Juno, a protein found only on the egg, and they bind, and in that single molecular moment a connection forms that changes everything as cell fusion occurs. The membranes merge into one continuous biological structure, and 23 chromosomes begin flowing into the egg's interior, carrying half of a unique human blueprint that has never existed before in the history of the universe and will never exist again. But the egg is not finished, because other sperm are still drilling, and if more than one fertilizes the egg the embryo cannot survive. So the moment Azuma-1 binds to Juno the egg fires an electrical wave, a surge of ion depolarization, that explodes outward from the point of sperm entry and races across the entire surface of the egg in milliseconds, hardening the Zona pellucida, slamming molecular gates shut, and making the fortress that 20 sperm were drilling through completely and permanently impenetrable in less than 1 second, ending the race with absolute finality. Simultaneously, the egg releases a massive burst of zinc ions that explode outward from its surface in a flash of light, the fertilization spark, real, measurable, and photographable, a phenomenon that stuns scientists when first observed, and whose brightness correlates directly with the developmental potential of the fertilized egg, as though life itself is announcing its beginning with light. Inside the egg now, the sperm's 23 chromosomes unpack and drift toward the egg's own 23 chromosomes, held in suspended animation since before birth, waiting for this exact moment, and they meet, they align, and from two incomplete halves one complete and unrepeatable genome is formed, 3 billion base pairs, the full instructions for building a human being, written in the language of molecules in a space smaller than the eye can see. 24 hours from now this single cell will divide, then divide again, and again, following instructions shaped by billions of years of evolution to become 2 cells, then 4, then 8, and in 9 months perhaps 100 trillion cells organized into organs, systems, thoughts, feelings, a face, a voice, a life. All of this from a single moment, a single molecular connection. A race run by 250 million and one by one, because life is not given, it is earned. Fought for at the molecular level in battles too small to see, and every person who has ever lived, every person alive on Earth today, began exactly like this, in darkness, in silence, with a spark.